Okay, can you all still hear me all right?
Yeah, we hear you, and
Yep.
we have your first slide as the only video we see, and
Great.
I think we'll go ahead and get started. I'll just remind everybody to mute yourself and thank Jim Gern for his willingness to participate and give us a presentation. I'll remind you all, the reason that we call these non-CME. It's not that they're any less quality than what we expect, and we know they're all going to be excellent presentations. It's just that our contract with the CME office ran out, and so they didn't uh, give us an extension for the talks that we're giving through June and July. And then we'll end our series in the end of July and resume in November later in the year than we normally will. So again, thanks, Jim, for getting this presentation together. You can see the title, Viruses Including COVID and Their Relationship to Asthma, from the other UW, as we call it out here, the University of Wisconsin. And go ahead and take it away. I'll remind you, everybody, mute yourself so you don't disrupt the presentation.
Great. Thanks, Len, and I appreciate the invitation to speak to your group this morning. So even though it's non-CME, I'm used to doing this, so I'll give you my disclosure. So I do consulting for MISA vaccines, who are developing an RSV vaccine, and then MedImmune, AstraZeneca, who also have some interest in antivirals, and then have some patents for rhinovirus production methods. But I'm not going to talk about anybody's commercial products on this at this time. So this is just an overview of how viruses influence asthma. And really, you see this relationship start in very early life, where we know that acute wheezing illnesses are oftentimes initiated, or almost always actually initiated, by viral infections. And we know now that the viruses probably start the process, but the background microbiome and changes to the microbiome probably add to these the severity of these illnesses. But in most kids, these go away and there's no sequelae. In some kids, they go on from wheezing illnesses with viruses to wheezing from multiple reasons and develop typical childhood asthma. These kids tend to wheeze with rhinoviruses. They tend to have allergen-specific IgE, and again, microbes probably play a role in this progression to asthma. And then once asthma is established, these same factors, so allergen-specific IgE, viruses, particularly rhinoviruses, and microbes, are very involved in acute exacerbations. And I'm really going to be talking about this part of the schematic in the next 45 minutes or so. So viral infections and exacerbations of asthma. We know that many exacerbations are initiated by a viral infection. And this is particularly true in young children. So in preschool children now, actually, most of the studies that we've been doing recently with molecular methods, you can find a virus 95% of the time. It depends a little bit where the child is located. School-age kids, 70 to 85%, and adults, half to 70% of the time, exacerbations have viral infection as a contributing factor. So in kids, it's particularly a strong relationship, and it was shown dramatically, I think, in this graph from the group in Canada, Neil Johnston and Malcolm Sears, who looked at Canadian school-age kids, and they took hospitalization rates, plotted them monthly for each year. You can see about a 15-year span there. And what they found was that there was a peak in asthma hospitalizations that was 17.7 days after Labor Day, every year. Labor Day varies by a few days. It's what school initiation is tied to. and you can see that it's linked very closely to these peaks and exacerbations. They tend to happen in the fall, and then a smaller one in the spring. You can see the little spring peaks here. And then the peaks for adults and younger kids lag by a few days, suggesting that the viruses are spread in school and then brought home.
Did you leave me any coffee?
So most rhinovirus infections don't cause exacerbations. I just showed you that they're very closely linked. But the bottom line is that most of the time, even with someone with asthma gets a cold, gets a viral infection, it's just a cold. And it's less than a third that go on to develop exacerbations in most populations. People with really severe asthma, you know, the relationships are, you see a little bit more of a percentage go on to exacerbation. And of course, the reason is that we're not totally defenseless against these respiratory viruses. We have neutralizing antibodies that moderate the severity of cold or prevent you from getting infected. There are interferon responses that slow down replication. And then the neutrophilic response is very strong, and these cells have some antiviral properties as well. So what are the risk factors for these, you know, 25 or 30% of colds that go on to develop exacerbations? And I'm going to talk about 3 today. Some risk factors related to the viruses, some related to allergy, and then some related to the airway microbiome, or bacteria is what I'm going to focus on in the airway. So let's talk about the viruses. So rhinoviruses are important triggers for exacerbations, especially in the fall and spring. In the wintertime, of course, we have a host of other viruses that can have similar effects. But those big fall peaks, most of the time it's a rhinovirus. And there's 3 different species of rhinoviruses and over 160 different types, serologically defined or genetically defined types. And any of these 3 species can cause asymptomatic illness or upper respiratory illnesses, and this is the most common outcome. But the A and the C viruses can also cause lower respiratory illnesses and wheeze. There are some structural differences between these viruses and probably some clinical differences between these viruses. The A viruses bind to receptors like ICAM1 and the low-density lipoprotein family of receptors, whereas the C viruses were recently found to bind to CDHR3, which is a cadherin-related protein. Not much known about CDHR3 until recently. CDHR3, again, it's a cadherin. We know that other cadherins like E-cadherin are involved in the strength of the epithelial membrane. And you can see the distribution of E-cadherin staining here in between the cells. It works like glue to keep these cells together. This is bronchial tissue, and these are cells grown at air-liquid interface with the same pattern. In contrast, this cadherin-related protein is on the apical aspect of ciliated epithelial cells. And so you can see a very different distribution, suggesting that it has a different function. And we still don't know a lot about what it does. There's some evidence that eosinophils can bind to CDHR3. It binds to itself. Its exact function is still not completely understood. But one very interesting thing about CDHR3 is that it's been identified as an asthma risk gene. And there's a single nucleotide polymorphism that's associated with early childhood asthma with severe exacerbation. So a particular phenotype where kids are well until they get a cold, and then they have severe exacerbations. So here's a schematic of what the genetic variation does to the structure of the protein, and then how that might relate to susceptibility to rhinovirus-C. So this is the tyrosine variant up above, and the variant down below is the low-risk variant. So the structure is different. The polymorphism is in this little red domain here, so it's in the 5th domain, and it probably has to do with how well this protein can fold. Actually, the more common variant isn't very well folded, and the expression on the surface of the cell consequently is lower. the asthma risk allele is folded a little differently, and more of it makes it to the surface of the cell. I'll just mention that humans are really the only ones that have this C529 variant. Every other mammal has this high risk variant. So there's actually evidence that in the last 5,000 years, the prevalence of this asthma risk allele has gotten lower and lower in the population. So what does this do to rhinovirus binding? Well, if you have more of the receptor on the surface, you have a little bit more binding, and then this is the log scale here. So at the end of 24 to 48 hours, you have about 10 times more virus in the culture in the cells that have the asthma risk allele. Is this clinically significant? The answer is yes. So we've done an analysis with 2 different birth cohort studies, the COPSAC study in Denmark, as well as the Coast birth cohort study in Madison. Here, what we're looking at is the risk ratio for Rhinovirus-C illnesses compared to an illness with any other virus that's associated with asthma, this asthma risk allele. And what you can see is that the CDH03 polymorphism increases the risk for developing Rhinovirus-C illnesses, but not illnesses with any other virus. So we know that rhinoviruses infect ciliated airway epithelial cells. What happens and what about that process leads to problems with asthma? So here we've just got a schematic, a ciliated epithelial cell. This is a secretory cell, and so forth. So Sarmi Basant is a postdoc in my lab who's working on a paper. I'm going to show you some early results here. These are cells that are cultured at air-liquid interface, and it just shows you what happens when you infect with rhinovirus. So these are no virus, and then infected for 12 hours, 24 hours, and 42 hours. I think what you can see is that by 24 hours, we see this nice ciliated epithelium up here. Everything looks fine. At 12 hours, there's something going on. And by 24 hours, you start to see some cells, some dead cells floating on the surface of the layer. And by 42 hours, there's a lot of dead cells. And you can see with this Alcian blue stain, a lot of mucin up here. So this reproduces, what you might think with a cold. After a few days of having a cold, you have a lot of mucin and a lot of dead cells floating around. It's often said that rhinoviruses aren't cytolytic or cytopathic, and that's wrong. You can see there's no holes in the layer here. It hasn't been wiped out because the epithelial layer can regenerate very quickly. But these are dead cells that have been killed by the virus. And in fact, it does have an effect on the barrier function. Finally, on the right here is just fluorescent staining, and it shows you that most of the virus is in these dead cells that are just floating on top. So this is what it looks like kind of at the immunohistochemistry level. At the single cell level, you can start to put together a story of how these cells coordinately respond to infection with virus. So it turns out this is a Tisney plot that looks at gene expression. We've got a few thousand genes that are being expressed here, and it puts it in a 2-dimensional coordinates, so you can look at groupings of cells. Turns out there's 4 different groupings of airway epithelial cells that are ciliated or developing cilia. Here's a group of cells here that are mucin secreting cells. These are pulmonary ionocytes, and these right here are basal cells and cells that are developing into secretory cells. So you can identify different groups. And so you can start to look at what happens with virus. And it turns out with this single cell technique, you can see virus inside cells as well, because the viral RNA looks like a human mRNA. So at baseline, this is an example of an antiviral gene, MX2. Here's a chemokine gene that brings in mononuclear cells. And it's pretty quiet. Here's the expression scale here. There's not much going on. 24 hours after infection, though, these ciliated epithelial cells are really lighting up with virus. A little bit of virus just kind of scattered elsewhere, probably just kind of sticking to the cells. But here's where the replication is happening. And you can see that some of these cells are mounting a muted antiviral response, and some are not. These cells are probably defeated. They've lost the battle, and you have high level viral replication going here. And interestingly, you start to see here that some of the infected cells are making some chemokine, but most of the chemokine, you think of this as the immune response to the virus, is coming from the cells that really aren't infected. So it's these goblet cells and secretory cells. You can see that as well at the 42-hour time point. The goblet cells and secretory cells are just lighting up. So that although they're not infected, they're certainly taking the lead in responding to the infection and calling in the cavalry. So mononuclear cells get recruited to the airway and help to rid the airway of virus. So the summary is that you have the ciliated epithelial cells being infected, and then as the infection progresses, they must be sending out some signals. We're looking to see what those signals might be. Certainly they make interferons, perhaps other activation signals. But what you end up with are, some of these cells dying and being shed with lots of virus attached. And then the nearby cells, some of them are protected by the interferon, and some of them are responding to the infection with chemokines and mucins. So I think this is going to be an interesting model. We're going to see, what happens and what's different in cells from asthmatic individuals versus non-asthmatic. We're anticipating more mucins, perhaps, and maybe a muted antiviral response.
Jim, this is Matt, and
So,
sorry if I missed it.
Hey,
So
Matt.
are the same cells, how are you? Good, thanks for joining us.
Good.
The same cells that are, the secretory cells that are generating the antiviral response there, are the same cells also generating sort of a mucus, hypersecretory response,
Yes,
or do
they
you see
are.
different populations? Okay, so
Yeah.
they can do both functions really at the same time.
Yes, and what's happening as well, Matt, is the proportion of the cells is changing as well. So ciliated cells are going down during infection, probably because they're being killed off. This is, we're putting a lot of virus in these cultures to do these kinetic studies. The basal cells also seem to slowly diminish, and we think that's because they're differentiating into new cells, probably more goblet cells and secretory cells, and the secretory population seems to be increasing. So along with making more mucin and chemokine and things like that, there's more of them as well. Okay, so how do these activities contribute to airway obstruction? And first of all, there's some data going back decades now that rhinoviruses grow, at least some rhinoviruses grow best at slightly cooler temperatures, so 34 to 35 degrees. Some actually, the C viruses grow just fine at 37 degrees as well. But when you measure temperatures in the large lower airways, actually in room air, breathing comfortably, your major airways are at 33 to 35 degrees. So the temperatures down here are just perfect for rhinovirus replication. And we've done inoculation studies where we found that in adults, when you inoculate in the nose with large droplets, about half the time, even in normal individuals, the rhinovirus infections extend to the lower airway. So, when you get a bad cold and you get that chest cough, this is a viral bronchitis, really, in many individuals. And then, I just showed you what happens at the single cell level. You can imagine if there's virus down here, you're getting shed cells, you have lots of inflammatory cells that are coming into the airway, you have oversecretion of mucins. This is going to lead to airway closure, exacerbate asthma. So I think are big contributors to what you see, and perhaps why it's not to bronchodilators all the time, with viral infection. Okay, let's move on and take a look at allergy and its role in promoting more significant rhinovirus illnesses and wheezing illnesses. And this is a great epidemiologic study that the Peter Heyman and his group at University of Virginia conducted, looking at allergy and rhinovirus-induced exacerbations. Case control study of kids presenting to an emergency department in Costa Rica, 96 with acute wheezing, 65 with stable asthma, and 126 non-asthmatic controls. Each of these kids had nasal sampling for PCR for rhinovirus and also had wheezing, IgE, allergen-specific IgE antibody, and FeNO measured during their ER visit. And dust mite is the major allergen in Costa Rica. It's a very humid environment. And not surprisingly, in kids who weren't infected with virus, you could see this nice relationship between IgE to mite, specific IgE to mite, and then the probability of current wheeze in this case control study. So the higher the mite-specific IgE, the more likely the kids were to wheeze. But just look what happens when you throw a virus in there. You know, you still have this relationship with mite, but it's really sky high. And the highly allergic kids who were infected with virus were almost all wheezing. So it suggests that allergy is working together with viral infection, you know, to promote these exacerbations. And this has led to speculation that allergy could inhibit antiviral responses. So is there something about allergic inflammation that impairs the antiviral response? And in the laboratory, these are 3 or 4 studies, there are about 5 more now that show a number of mechanisms. So there are transcription factors induced by allergy that suppress interferon. Eosinophilia suppresses TLR7 responses. This is one of the big hub genes in the anti-rhinovirus response. And then activation of the high affinity IgE receptor suppresses antiviral interferon responses. And this is some work that Michelle Gill has done, and it's been verified in a couple of different laboratories. So it leads to the question of whether neutralizing anti-IgE with omalizumab could reduce the severity of viral illnesses and reduce exacerbations. And I'm going to show you data from 2 studies conducted by the Intercity Asthma Consortium showing that omalizumab added to guidelines-based therapy in children with moderate to severe asthma did indeed have effects. And the 2 studies were ICATA, which used year-round treatment with omalizumab, and then a follow-up study that treated kids for a couple of months before school started to see if they could reduce that big fall bump in exacerbations. So here's the main data slide for the PROS study. This was year-round treatment with omalizumab. The kids who didn't get omalizumab had guideline-based therapy. And what you can see on this graph here, it's looking at the percentage of kids with exacerbations, is that exacerbations were lower in the omalizumab group than in the standard treatment group. But what really stands out is that the seasonal increases in exacerbations that are mostly, as we know, due to viral, or initiated by viral infections, were really blunted in this study. So overall, 25% less days with symptoms, reduced exacerbations, so 30% and 49%, and this big effect in seasonal exacerbations. Well, here's the follow-up study, the PROS study. During the run-in period in the spring, you can see that both kids had similar levels of exacerbations. Then they were randomized for treatment with omalizumab or standard care, and then you can see that the omalizumab kids just didn't have that fall bump in exacerbations. So it turns out that 85% of exacerbations were associated with virus. There was about a 50% reduction in exacerbations. And blood had been taken from these kids before and after randomization. And what they found was that if you looked at interferon responses in the blood cells, so they were incubated with rhinovirus in the laboratory, the kids who had more interferon production, you know, after getting omalizumab were very unlikely to develop exacerbations. So it suggests there might be some sort of an antiviral effect. You know, there's no direct antiviral effect of omalizumab reducing viral replication, but you know, it certainly could work through a number of inflammatory mechanisms. So as part of the PROS study, there were weekly nasal mucus samples that were analyzed for virus. 6,000 specimens in all. And this was combined with symptom diaries. And they analyzed effects of omalizumab versus placebo, which was standard care. And interestingly, with these weekly specimens, you were 30% less likely to find rhinovirus in kids who were treated with omalizumab. And viral colds, as shown on the graph here, went down by about a third. And there was evidence of some sort of an antiviral effect because both viral shedding, which went down by about a day, and peak viral shedding, which went down by about a log unit, were significantly affected. And these are pretty close to what some of the antivirals have been shown to do. You know, you think of pleconaril and so forth. You know, cut a day or two off of the cold and half a log to a log of viral shedding. So in summary, you know, omalizumab, which it has a fairly focused effect at you know, blocking the ability of IgE to attach to cells and reducing the surface expression of high affinity IgE receptors on cells, reduced rhinovirus detection, reduced the duration and quantity of rhinovirus shedding, and reduced rates of colds. So obviously, this is not a practical treatment for the common cold, but I think what it does is it gives us proof of concept that if you reduce allergic inflammation in the airway, you can improve people's outcomes with viruses, with rhinoviruses. Okay, so let's go on to bacteria. And you know, we've known for years that there are interactions between rhinovirus infections and bacteria. Rhinovirus infections have been linked to subsequent bumps and pneumonia. Certainly they can initiate a bacterial sinusitis, and in kids it's been known for years that viral infections are usually the trigger, a bout of otitis media. So this led us to ask whether rhinovirus bacteria interactions contribute to the severity of colds and exacerbations of asthma. So to approach this question, we recruited a little over 300 kids, about half with asthma. These were school-age kids who had weekly nasal mucus samples during the September cold season, and then again in April during the cold season. We looked for common respiratory viruses, and we typed the rhinoviruses with partial sequencing, and then used PCR to also quantitate the common bacterial pathogens, so H. flu, strep pneumo, and Moraxella catarrhalis. The kids kept daily symptom diaries for both cold symptoms and asthma symptoms, and we had a definition for an outpatient asthma exacerbation as having at least moderate symptoms, and the criteria for increased albuterol use or a 20% reduction in peak flow. And one of the things that was quite evident is that virus preceded the detection, the increased detection of bacterial pathogens. I'm showing you the data for strep pneumo is really the same for Moraxella and for H. flu. What really mattered was what happened the preceding week. A virus week before, you know, these kids were much more likely to have strep pneumonia and to have a lot of it. And interestingly, we know, remember, the A and the C viruses are the ones that are associated with exacerbations, and the B really aren't. You know, these mostly cause mild colds or asymptomatic infections. And when you look at the quantity of bacteria like Moraxella, the A's and the C's led to increased Moraxella, whereas the B viruses really didn't. So this suggests a model where during a respiratory illness, viruses come in, and in kids who have, you know, low levels of these pathogens, they can proliferate, and you know, probably the virus primes the lower airway somehow, bacteria, and it's this combination, this sequence that is related to, you know, more significant illnesses. So we tested that in this study. We looked at colds that were associated with Moraxella only. There were very few of these. Well, this was actually weeks where you detected Moraxella only. and no virus. And your odds ratio for having an illness was not significant. In weeks where kids had rhinovirus only versus no pathogen, you saw a bump in the risk ratio for, the odds ratio for illness. But kids who were most likely to be sick were the ones who had both the rhinovirus and Moraxella. And then you can see here that rhinovirus and Moraxella, you're more likely to be sick than if you had just rhinovirus alone. And you see the same general pattern for exacerbation. You know, half of our kids had asthma, and so the confidence intervals are a little wider, but really the same sort of pattern, suggesting that these bacteria are working together with the viruses to cause symptoms of illness and exacerbations. So we're starting to look at this in the laboratory. You know, what happens when you put Moraxella and virus together in a culture of airway epithelial cells? And we were surprised initially that if you put Moraxella in a culture of airway epithelial cells, as we put in 1 × 10⁷ Moraxella, within 48 hours, you see that 99% of the bacteria have died off, and the cells were perfectly happy. We thought we would get bacterial overgrowth and all the cells would be dead, but they're not. And actually, half of kids out there have Moraxella in their nose, of young kids. So it's not that surprising that they can peacefully coexist. But if you have a virus, so if you co-infect with rhinovirus, you get, you know, a great enhancement in terms of the amount of bacteria that are left. And actually, they're all cell associated. So, so bacteria somehow leads to these, a viral infection somehow leads to Moraxella becoming cell associated and surviving in the nose. There's no effect at all on rhinovirus replication. You know, this is the amount of virus at the end of 48 hours. We put 1 × 10⁷ in, and you know, there's 1 × 10⁶ in, and there was vigorous viral replication, but no evidence that it was affected by bacteria. So we're continuing to look at this and wondering how this cell-associated bacteria might influence inflammatory responses. So anyway, to sum up this part of the talk, the epidemiology of asthma exacerbations, you know, we know there are environmental factors that are important for exacerbations and host factors as well. This is clearly true for virus-induced exacerbations as well. We talked about how allergy, and I didn't show you the data on allergen exposure, but these certainly work together to enhance your risk of virus-induced exacerbations. We know the type of virus makes a difference, and the A and the C species viruses are most likely to cause exacerbations. And then there are bacteria, especially Moraxella, that seem to be closely associated with these acute severe respiratory illnesses. And of course, a number of other factors that are related to the host, the genetics of the host. We talked about CDHR3, baseline inflammation before you get a cold, what your lung function is before you get a cold, and of course, adherence to anti-inflammatory medications. All of these affect exacerbations. And I think this paradigm, you know, can help us to think about specific treatments for this. You know, right now we don't have antivirals, but we certainly can treat inflammation in the airway. And there are a number of studies going on now. I showed you one with omalizumab, but seeing as to whether IL-5 mediated inflammation, eosinophil mediated inflammation, you know, with dupilumab, are these all going to have effects on the epithelium that lead to better resistance against viral infections? And of course, there are potential therapies here: biologics, immunotherapy, inhaled corticosteroids, and allergen avoidance. You know, these all have a good rationale to prevent virus-induced exacerbations. For viruses themselves, we know that there are certain viruses that are more likely. We also know that the immune response to virus, especially interferon responses early in the illness and neutralizing antibody responses, you know, could be a role for vaccines here, but developing a vaccine to rhinovirus is a very tall order. We'll talk about that in a minute. So antivirals, TLR agonists, which may generally increase the resistance of the airway to viral infection, and then vaccines. And then bacteria. The natural resistance to bacteria comes from neutralizing antibodies, Th17 responses, and of course we have to think about pathogens versus commensal bacteria. And there could be a role in the future here for vaccines to inhibit specific bacteria. TLR agonists, again, that boost antibacterial responses. Probiotics that could have beneficial effects on the airway epithelium and also keep the pathogens at bay. And, antibiotics have a potential role. We know there's a big downside to antibiotic use, and so we'd like to minimize this for use in exacerbations unless we know there's a high-risk group that would really benefit, and that needs more study right now. And then one thing to think about that's important is that any of these inflammatory mechanisms lead to mucin hypersecretion. Bacterial infections do, viral infections do, and allergens do. And so this may be a common target. And of course, there's a lot of efforts going on now to try to understand the genes that regulate mucin hypersecretion. It's not just IL-4 and IL-13. There are other mechanisms as well that mediate the effects of viruses and bacteria here. So I'm going to finish with a couple of slides, a few slides on vaccines, and then a few slides on COVID, and then we'd be glad to have some discussion here. So what about vaccines? Well, the opportunities here are that we know that naturally occurring rhinovirus antibodies are neutralizing. They are protective. You have a substantial natural protection that starts, within a few weeks of your infection and lasts from we don't actually know how long it lasts, 2 to 3 years in some of the older studies, maybe longer for C viruses. And then we have at-risk populations that are recognized. So highly atopic kids are very likely to wheeze with viruses, kids with CDHR3 variants, and then of course people with more severe asthma, COPD, and other chronic lung diseases. The challenges are that there's 180 serotypes. We can cut a few of these out because the B viruses are attenuated. And the other challenge is that natural immunity has limited cross protection. So if you're immune to one of these viruses, A104, let's say, it might give you a little bit of immunity to some of the very closely related viruses, but still, there's an awful lot of viruses to consider. This has led to some new approaches to a rhinovirus vaccine. Some of the capsid proteins have some cross-reactive epitopes, and so this has been looked at. There's some cross-reactive T-cell immunity. T-cell immunity seems to give you fairly weak protection against illness in the models that have been studied so far. And then the idea that you might be able to develop a multiplex vaccine. People have always looked at the 100+ rhinovirus and say, oh, you could never make a vaccine to that. But we have a pneumococcal vaccine that has 23 valent antigens. What's the limit? And so Marty Moore, and we helped him a little bit with this study, had developed a 50 valent vaccine when he was at Emory. This went through some preclinical trials. First, he vaccinated mice with a 25 valent vaccine, got good antibody responses, and then macaques with a 25 valent and a 50 valent RVA vaccine. It was an initial vaccine with a boost on day 28. The neutralizing antibody titers were measured on day 46. 2 to the second, or a neutralizing antibody titer of 4 is generally recognized as being protective. And he got an antibody titer of greater than 4, actually much greater than 4, to 49 out of 50 viruses. So this vaccine would be difficult to make, but you can put 50 viruses together and, at least in animal studies, get good neutralizing responses. Don't know how long this lasts. It hasn't been studied yet. Yury Bochkov, my good colleague here at Wisconsin, has recognized the potential for development of a Rhinovirus-C vaccine and has a grant to look at this now. And we can identify high-risk kids who are genetically at risk, certainly associated with wheezing and asthma, maybe associated with asthma development. And there are a smaller number of Rhinovirus-C types, which appear to be very immunogenic in the studies that we've looked at so far. So could a vaccine be developed? He's working on this now. And actually, he put together, these viruses are tougher to make, but he had a 9-valent vaccine here with an initial priming vaccine and then a boost. And you can see even with just one priming vaccine, he got very good titers to 9 out of 9. So he's looking at how many you would have to put in, to get immune, to become immune to all 60, and we'll see how that work turns out. Okay, and finally, I just want to say a few things about COVID-19 and asthma. Again, showing you information about viral infections can cause severe lower respiratory illnesses. The CDC mentions moderate to severe asthma as a risk factor for severe COVID-19 illnesses, but the clinical support for that statement is fairly weak. So reports on whether asthma is really a risk factor for COVID-19 hospitalization have been mixed. Most of the stuff in China really didn't find that. Some of the US reports show some evidence that there are more than expected people who report asthma, but most of those people also have other risk factors. And so Castro Rodriguez and Eric Forno recently put together a review in kids, and there's almost no data in kids to even assess this. So we don't know, precisely how much of a risk factor it is. Here's just a little snippet of SARS-CoV-2 molecular virology. It's an enveloped single-stranded RNA positive strand virus. It's got the biggest single-stranded genome of any virus. And the positive strand means that this protein's right away. It has a spike protein that attaches to the ACE2 receptor on the surface of ciliated epithelial cells. And this TMPRSS2 is a protease that primes the spike protein for receptor binding. And so you probably have seen this data because Matt did this analysis here. This is work by Dan Jackson and Matt Altman and some of us at the Intercity Asthma Consortium, showing that allergy seemed to be inversely related to ACE2 expression. So we have a birth cohort in 4 inner cities around the country. And when these kids were 11 years of age, nasal epithelial cells were obtained by brushing. And then Matt did an analysis of gene expression where he compared ACE2 expression to allergy and asthma. And you can see the 4 groupings down here, yes or no for asthma and for allergic sensitization. What you can see is that this group who had sensitization plus no asthma, or the group that had asthma and sensitization, had a lower expression of ACE2. And then when you look at the degree of sensitization with asthma, so low, medium, and high, you can see that the kids who have asthma who had high level allergic sensitization really had a down that allergic sensitization by downregulating ACE2 might protect these kids somewhat against having more severe COVID infections. So a second study came out and verified this effect on ACE2, but also looked at this TMPRSS2 gene that primes the spike protein. And what they found was that allergy seemed to be associated with an increase in expression of this gene. So you have less ACE2, but you have more of the protease. How does that balance out in terms of your risk for COVID? And right now that's still speculative. And there's been one other analysis of this. This was by the SARP group, the Severe Asthma Network group. We looked at 356 patients, 88 healthy, 125 mild to moderate asthma, 143 with severe asthma. These were all adult patients who had bronchial brushing, and then ACE2 expression data. And you can see there just isn't any effect at all of asthma on expression of either ACE2 or this protease gene. However, they did also look at the relationship between expression of airway inflammatory genes and ACE2, and they found a significant positive relationship, although it's a little bit loose, you know, for Th17 to promote ACE2 gene expression and for Th2 gene expression to be inversely related to ACE2, again, suggesting that allergy, you know, might downregulate receptor expression. So anyway, these are all really interesting mechanistic studies. Obviously, we're all really interested in how ACE2 gene expression regulation would relate to the severity of illnesses. Right now, there have been some reviews of COVID-19 and biologic therapies. And right now, unfortunately, there's just no evidence on how biologic therapies influence COVID-19 outcomes. We know they do tremendous, you know, have tremendous potential to control asthma. As to what they do with COVID-19, we just don't know. So, you know, it's been suggested, should we really be neutralizing IL-4 and IL-13 if they're suppressing, you know, the ACE2 receptor? But, you know, all of the big you know, guidelines organizations have stated that in the absence of evidence of harm, the expert opinion is to continue biological therapy when indicated for asthma, because we know it does good, you know, for asthma itself. So I'd just like to close and acknowledge my colleagues at the Institute of Molecular Virology and Carol Ober at University of Chicago, who did the genetic data, the group at UW. And I should have put an acknowledgement for Matt Altman at the other UW, because yes, folks, we were the first UW here in Wisconsin who helped with those gene expression studies. So thanks very much for your attention, and I'd be glad to address any questions you might have.
Go ahead, folks, and unmute yourself if you have any questions. Thank you, Jim, very much. This is sort of a naive statement, but it seems to me counterintuitive that our immune system would be set up that if you're an allergic person, that mutes your viral immunology, your viral immune response. That would be an unhealthy way for humans to evolve.
Yeah, I think it is unhealthy, and you know, we just see this very close relationship between allergic inflammation on one hand and the response to viral infections in general on the other. You know, the COVID-19 may act differently. We don't really know. But I think the bottom line is that epithelial cells do really well handling insults. They have trouble when they have to handle more than one insult. So they do pretty well with bacteria. They do pretty well with viruses. They do pretty well with pollutants. And they do pretty well with allergic inflammation. But you start throwing more than one at them, and you start to see things fall apart. So, for example, we know that allergic inflammation also reduces the ability of a cell to handle oxidative stress. Those mechanisms get downregulated. And I think, you know, a cell just has so much energy, and, you know, so many ways it can regulate itself. And so I think those combinations are where you really run into big problems.
You got other questions out there?
Is there a word of the day for?
Oh, we're not getting CME credit, so there's no word.
Okay. Okay.
There, one other thing, I was reading something in the New York Times about the size of respiratory particles in a humid and a non-humid environment. And, I've always thought that the reason we have a bump in infections after Labor Day, as you showed, had to do with kids going back to school and being adjacent to one another. But it also pointed out that when you turn on the heat in a house and the relative humidity drops, particle sizes get much smaller and carry in the air much longer. And that the implicit suggestion would be that hence viruses are more communicable because of that. Comment on that?
Well, that's an interesting theory, and the epidemiology of rhinovirus infections have been studied fairly intensely in single centers, but it's really interesting when you start to look at globally, what happens. So fall and spring are the big time periods for rhinovirus infections. These infections, as opposed to flu, never really go away in any single location. But actually, in tropical areas, there have been some studies in Brazil and Africa and so forth. They don't see such a big fall and spring seasonality. And so it may be because the climatic conditions are more stable, or at least whatever is conducive to rhinovirus spreading is more stable. And maybe that has to do with humidity. I don't really know, but it's interesting. Rhinovirus is spread both by aerosol and by contact means. They don't spread, their R0, I guess, is quite a bit lower than what you see for either flu or COVID. So they're not as contagious. It's just that there's so many viruses present in a community at any one time that, you get lots of exposure.
Yeah, this was written in the context of concern about COVID and, if you will, a second wave in the fall, although we're obviously not even anywhere near our first wave. So anybody else out there with questions? I want to thank Drew. This was the first Zoom session, as I mentioned, without administrative support, and worked perfectly.
It's called dumb luck.
Well, Jim, thanks a great deal. Again, I'm pretty sure we got this recorded for a broader audience to listen to it. Another friend and colleague will be talking next week, for those listening to me, Steve Durham from London. So we'll see if we cannot screw up that presentation.
All right. Thanks, Drew. Thanks, Len. I really appreciate the invitation. It's been a pleasure to speak with you today. I wish I could have been there. I've got I have a son that some of you may know, and a granddaughter coming in the next month or two. So I'm delighted to visit in the future.
Well, I hope the time comes that people can get on an airplane. We can have you out here. As I said, we're going to delay the start of our traditional series till November. And part of my thinking there is I'm hoping by November people can, or subsequent shortly after that, can travel, and we can have you out here in person.
Right. Very good. Thanks again, everyone.
All right. Bye-bye.
Bye-bye now.
Thanks, Jim.
You're welcome.
Paul McBride, I see you up there. How are you? Let me see who else is still online here.